Welcome dear students. This is my cellular energetics lecture series. In this video I will going to talk about glycolysis. But at first I will give you some basic information so that you can understand this topic easily. Cellular energetics means how cells acquire energy, because life requires energy. To do anything it requires energy. Now the cellular energetics can be divided into 4 stages. So there are 4 stages. I can write here stage 1. This stage 1 is the glycolysis process. We will learn this topic, glycolysis. The stage 2 is known as tricarboxylic acid cycle or citric acid cycle or Krebs cycle. So I am writing here only TCA cycle. In the stage 3, this is very important stage in case of cellular energetics, that is known as electron transport chain. In short we call ETC. Now by this electron transport chain a phenomena occur. We call it generation of proton motif force. This is very important, you remember, this point is very important. PMF, proton motif force. By electron transport chain it produces a phenomena called proton motif force. Now at the stage 4, the last stage of this cellular energetics, that is generation of ATP. So ATP production or ATP generation. So if I divide this cellular energetics lecture series, we can found that there are 4 stages. The first stage is glycolysis, second stage is TCA cycle, then is electron transport chain, then the production of ATP by an enzyme called ATP synthase. Now the important point about this, the proton motif force actually helps in the generation of ATP, and this process is called chemiosmosis or chemiosmotic coupling. So I can write chemiosmotic coupling. This is very important process. And do you know that this production of ATP is also known as oxidative phosphorylation. Phosphorylation. Oxidative phosphorylation. This is different from substrate level phosphorylations. I will talk about in more details. So basically all these 4 stages are actually known as aerobic respiration. Aerobic respiration. These all 4 stages is actually aerobic respiration. Now there is another way by which cell can produce energy. We call it photosynthesis. So in this total series I will talk about all these things in more details so that you do not miss any topic or any point for your preparation. Now today's topic is glycolysis. The name glycolysis come from glyco means sugar or sweet and lysis means breakdown or split. So sweet split or breakdown of sugar. Here glucose is the starting molecule. Glucose. We all know that glucose is a 6-carbon molecule, monosaccharide. At the first stage of this glycolysis, glucose is actually converted, very subtle conversion occur. That is, it actually modified, this glucose is modified to glucose 6-phosphate. Only a phosphate group is actually added at the carbon 6 positions. Now I will tell you some basic information about glycolysis also. This glycolysis process is actually occur in the cytoplasm. You have to understand this. Cytoplasm. All these stage 3, stage 2, stage 3 and stage 4 occurs in the mitochondria. This glycolysis process occurs in the cytoplasm. And cytoplasm. And all the enzymes are present in the cytoplasm that are water soluble. There are 10 enzymes are responsible for glycolysis. Among these 10 enzymes, 3 enzymes are most important because they are called rate limiting enzymes. Rate limiting, that actually maintain the total process. 3 enzymes are rate limiting enzymes. Because why they are called rate limiting? Because they are allosterically regulated. They are allosterically regulated. So it is called rate limiting enzyme. Now the first step, glucose to glucose 6 transformation, occurs by an enzyme called hexokinase. So this is the first step of glycolysis. Now I have right here one symbol that is like this. In glycolysis I will draw another symbol like this. This type of symbol is called irreversible reaction. Once the glucose becomes glucose 6-phosphate, it cannot convert it into glucose. This is called irreversible reaction for this enzyme. But many enzymes can perform reversible reaction. So in the glycolysis there are 3 rate limiting enzymes are present. They actually go through irreversible reaction. So that means the first step of the glycolysis here, it is actually this enzyme is a rate limiting enzyme, hexokinase. So why the first step of this glycolysis are so important for the whole glycolysis process? Now the reason is, suppose this is a cell. Okay, so how glucose enter inside a cell? The answer is there is a uniporter called GLUT, GLUT protein or GLUT protein, GLUT, glucose transporter. Now this glucose transporter helps in the transport of glucose inside the cell. In my GLUT transport lecture I have mentioned that this GLUT4 actually have 12 transmembrane alpha helix, and in that video I have also mentioned that this GLUT protein actually has, or all uniporter proteins are actually in pseudosymmetric in nature. That means let's suppose the GLUT protein actually have 12 transmembrane alpha helix, but let's suppose there are 10 alpha helix. Let's suppose these are the alpha helix, there are 5. Now pseudosymmetry means the structure of the protein is like that. Let's suppose if this hand is look like this, then the other hand will be like this, other hand. So the main fact about this pseudosymmetry is that through this pseudosymmetric nature of this protein, let's suppose this is a GLUT protein, through this pseudosymmetry, let's suppose there are 6 alpha helix, 2, 3, 4, 5, so 6. Let's suppose there are 6 alpha helix, there are another 6 alpha helix will be like this. So if this portions helps in the entry of glucose, then the half pseudosymmetric region also helps in the out of this glucose from the cell. You have to understand this. GLUT protein or GLUT protein can help in the entry of glucose inside a cell. Also the glucose can come out from the cell to the outside by the help of GLUT protein, because it has pseudosymmetric structure, so that bidirectional movement can occur. It can enter, it can come out, but a cell requires glucose all the time. Now this glucose should be trapped. So once a glucose enter inside a cell, it should be trapped. So this is called trapping of glucose. So this process is so important, and this is called rate limiting enzyme. So hexokinase is a rate limiting enzyme. In the first step it is called trapping of glucose. How this trapping occurs? This glucose, when it is added with a phosphate, the phosphate is actually negatively charged ions, so molecule. So this phosphate group makes this glucose negative charged molecule, and any charged molecule cannot pass through this transporter. So by this way, it actually make the glucose impermeable by this transporter. So this is one step, first important step. I can draw the structure of a glucose so you can understand. Let's suppose so this is actually basically a structure of glucose. I can write the carbon 1, 2, 3, 4, 5 and 6. Now what happens here? You can see the glucose 6 position is actually added with a phosphate group. So this hydrogen actually removed, a phosphate group actually added. So this oxygen actually remains negatively charged, so that this molecule, this glucose 6 phosphate, because this is a 6 carbon molecules, it actually gets negative charge, and any ions or any charged particles cannot move through uniproton. So by this way it actually traps the glucose. Now, important fact about the hexokinase enzyme. The name of this hexokinase refers hexokinase. Hexo means hexose, that means any 6 carbon sugar molecules. There could be different 6 carbon sugar molecules. Not only glucose is a substrate for hexokinase. Yes, there are many isozymes or isomers of these isoforms of this hexokinase called hexokinase 1, 2, 3. And there is another hexokinase I am writing in this lowercase, that is hexokinase 4. Yeah, this hexokinase 4 is different from 1, 2, 3, so I am writing separately. Now this hexokinase 1, 2 and 3 can convert not only glucose, but it can convert or change like fructose, mannose. You have to understand this. This hexokinase 1, 2 and 3, they can convert this glucose, fructose and mannose, but hexokinase 4, specifically known as glucokinase, it can convert only glucose. So glucose is only substrate for hexokinase. Hexo means 6 carbon. Now another important fact, you can ask me, where from this phosphate group actually generated? Yes, the answer is here. ATP is actually destroyed to make ADP, and we know there is adenosine, that is called triphosphate. That means there are 3 phosphate. One is called alpha, beta and gamma. This gamma portion of this phosphate actually transfer. So this is the gamma phosphate. Now, yeah, another thing is, you can remember magnesium actually associated with this ATP molecules, magnesium. Yeah, now I can write here a very important fact about this, just this first step. So I am giving you the maximum information that you require. Hexokinase 1 is present, hexokinase 1 is present in all tissue cell, body's all tissue cells. Okay, hexokinase 2 is present specifically heart and muscle cell. Hexokinase 4, or I can write glucokinase or hexokinase 4, is present in the parenchymal cells of liver and the beta cells of pancreas. Do you know there is a huge difference between this set of enzymes? They are isoenzymes, but there are so many differences. I have mentioned that this hexokinase is under the 3 rate limiting enzymes. They are controlled by allosterically. Now I can write here that the hexokinase, let's suppose this is a hexokinase. This hexokinase has a substrate binding site. Let's suppose this is a glucose. In case of 1, 2, 3, it could be glucose, fructose and mannose, but in case of hexokinase 4, glucokinase, it's only glucose. So there is a binding site for glucose. There is another binding site for ATP because it is also a substrate for this enzyme. But there is another important allosteric site present inside the hexokinase that is actually bound by the product glucose 6-phosphate. You have to remember this. Glucose 6-phosphate bind with this site. Now there is a difference. Hexokinase 1, 2, 3 have this allosteric binding site and can be inhibited. So this glucose 6-phosphate actually inhibit this hexokinase. So this can inhibit this enzyme. Now as this hexokinase 4 don't have this type of binding site, so it is not actually inhibited by glucose 6-phosphate, but it is actually inhibited. This glucokinase is inhibited by fructose 6-phosphate indirectly. You remember this, indirectly it can inhibit. So there is a difference. This is the first difference of these two things. Yeah, I have mentioned that only the substrate is glucose, and there is the, so this is the second difference between these two set of enzymes. There is another important difference between these two enzymes. So that we have said that glucokinase is present in the beta cell of pancreas. It acts as a sensor. I have discussed about that GLUT2 also acts as a sensor, and there is a similarity between this hexokinase 4 or glucokinase and the GLUT2. But how? You can see here that hexokinase 1, 2 and 3 have Km value. Km means Michaelis constant value, or you can say the affinity towards the substrate. Km value of 1, 2 and 3 is so low. I can write 0.002 km, let's suppose. Then this hexokinase 4 Km value or glucokinase Km value is 5 millimolar. Okay, Km value. So this Km value is so high for hexokinase 4. That means, simply means, these 3 molecules, 3 enzymes has higher affinity towards the substrate. The lower the Km, the higher the affinity. The higher the Km, the lower the affinity. That means this hexokinase 1, 2, 3 have higher affinity towards the substrate, but this hexokinase 4 or glucokinase has lower affinity. Approximately I am telling you 250 times, because you can see here the difference between 250 times of the Km value. So the affinity towards the glucose would be 250 times lower in case of glucokinase. Now you can ask me why then it acts as a sensor. It has low sensitivity or low affinity towards the subject, but it acts as a sensor. The reason behind this is a trick cells play. That is, something that actually have lower affinity, that means when the substrate concentration becomes so high, then this this enzyme can be activated. You have to understand this. Say that 20 glucose can activate these 3 enzymes, then activate, but this 20 glucose cannot activate this enzyme. But if there is 2000 glucose molecules, then these 3 enzymes also activated, but then this enzyme will get activated. this hexokinase 4 enzyme will get activated because it has low affinity. So the number of glucose should be high. So when the glucose level increases in our body, glucose kinase actually gets activated. And this glucokinase present in beta cells of pancreas that helps in the secretion of insulin. And do you know, it also regulates another enzyme in the alpha cells of pancreas called glucagon secretion. So glucagon. So this enzyme actually acts as a sensor. So in this video, I have only talked about the first step of this glycolysis. In the subsequent classes, you will learn the glycolysis. So as the classes will go on, I will tell you the all 10 steps in very details so that you will not miss any informations. So hope this video will help you. Thank you for watching and always keep learning.